Hello everyone, welcome to another learning session with online biology tutorials. If you haven't subscribed to my channel yet, kindly do so and do press the bell icon for the recent updates. I hope you will be benefited by my tutorials. Please do let me know through a comment in the comment box. It will be highly appreciated. Now in this video we are going to solve a problem, and the problem says if the coding strand of DNA has the sequence as A, T, G, C, T, A, T, G, find the sequence of the template strand and messenger RNA, right? So here what we need to do is we need to know what is coding strand of DNA, what is template strand of DNA, and how is messenger RNA formed from DNA. So let's start. We know already that double helical DNA, it has two strands. One runs from 5' to 3' and another is 3' to 5'. Whenever you are writing these DNA strands, just do remember that arrow should always come near 3' end, because this is a rule. So this is our 5 to 3' end at the top, and at the bottom we have 3' to 5' strand. Now remember, this is a rule that the 5 to 3' strand, it is known as the coding strand, and it is also known as the sense strand. Naming is very important, whereas the other strand, which is anti-parallel, it is known as the template strand, and the other name of the template strand is anti-sense strand. We will discuss this as well. Why are they known as sense and anti strand? But first we will concentrate upon DNA base pairing rule. Now here they are talking in the question about the coding strand, right? So coding strand means the above one. So we will write the bases in it first, okay? So first base given is A, then T, then G, then C, then T, A, T and G. This is what we have done as the initial step. Now our main job is to know that as per the base pairing rule, A always binds to T with double hydrogen bond and vice versa, right? And what else we know? We also know that C binds to G with triple hydrogen bond and vice versa. Only this thing we have to keep in mind for solving such questions. This is the thumb rule, right? Now let's come on and solve it. So here the first base is A, so we will write T. The second base here is T. We are going to write A as per the base pairing rule. Third one is G. We are going to write C. Then the next base is C. We are going to write G. Next base is T. We are going to write A. Next base is A. We are going to write T. Next is T. We are going to write A. And then last base would be C, because on the above strand it is G, and as per the base pairing rule we have seen that G always binds to C. Now we will make the hydrogen bonds. Here it will be 2, 2, 3, 3, 2, 2, 2 and last 3. So that means we are done with the first answer, okay? Because they were asking about the coding of the template strand, right? So here this is our template strand and we have found out the coding. So template strand is always 3' to 5' and the sequence in it would be TAC, GAT, AC, right? We are done with our first part. Now let's move ahead. So what are we required to do now? We need to find out the sequence of the messenger RNA. Okay. Now to understand it, let me draw the strands once again. This is our 5 to 3 prime end. You have already understood that this is coding strand. The bottom strand, it is the template strand. We have also discussed about it. That means the two strands of DNA, they are anti-parallel to each other, means the base pairing rule will be followed, and that's how we do the bases, right? Now another important thing, this strand would be the one, means template strand would be the one that would be serving as a template for the synthesis of the messenger RNA. Right? In the very first video, we have discussed about central dogma. If you haven't watched that video on DNA structure and function, do watch it. Right? So the formation of RNA, messenger RNA from the DNA, this is known as transcription. And we know that template strand would be utilized for this purpose. Now this RNA, this is single stranded. DNA is double stranded, right? Now another important difference which you must know is that DNA is also a polynucleotidic structure, means it would be containing base, sugar and phosphate, right? We would be looking in a separate video where we'll be working about DNA and RNA differences, but right now for this question you must understand the basis difference, right? So purines are A and G in the case of RNA, but there is a difference in pyrimidines, right? There is a difference in pyrimidines, and there is no thymine in RNA. In spite of that, we have uracil and the cytosine is there. So in place of thymine, we have another base which is capital U or uracil. We just need to keep this thing in mind, right? Another important thing which I am telling you a trick, that the sequence of messenger RNA would be the same as the coding strand, right? So we will first write the coding strand, and this will make our task easy. So the coding strand has the sequence of A, T, G, C, T, A, T and G. Right? We would be working out on two ways to find out the sequence of messenger RNA. First, I am taking the coding strand and I will help you out how to find the sequence of bases in the messenger RNA, right? So we know that messenger RNA will always be in the direction of 5 to 3 prime because it is anti-parallel to the template strand, and you can see here 3 prime end is there. So here it is just the opposite, 5 prime. Here it is 5 prime. So here it is anti-parallel. I have written 3 prime. Okay. So right now we are deducing the sequence of bases using a coding strand. Okay. So this is our messenger RNA, and M is always written as small and R and A will all be in caps on. Okay, so what you need to do is, here A is there, so you will write A. Okay, here T is there, there is no T in RNA, you will write U. Rest, G is also found, C is also found, again T is there, so you will be writing U. Again A is there, which is also found in RNA, again U has to be written because no T in RNA, and G will be written as such. So this is our sequence of the messenger RNA which we have deduced using coding strand. So coding strand sequence will be same as that of the messenger RNA. The only difference would be T in the case of DNA will be replaced by U in RNA. Okay, let's find out the other way. If they have given you the sequence of just the template strand and they are asking you to work it out, so we know that coding strand has been given to us. So this is our coding strand. Let's suppose it is A, T, G, C, T, A, T, G. Fine. So this is our coding strand. Let's draw the template strand of DNA with another color. right? This is our template strand, and you know that they will be following the base pairing rule. So here it will be T, A, C, G, A, T, A and C, right? I'm not drawing the hydrogen bonds properly because I have already explained above. Okay, now what we need to remember, template strand of DNA, right? This is our template strand of DNA. Messenger RNA is just the anti-parallel to it, so we will be drawing it with green color. This is our messenger RNA. Now bases will be opposite to each other as per the base pairing rule, right? So here you can see that here it is T, right? So here we would be writing A because T always binds with A always binds with T, but there is no T in RNA, so we would be writing U. Here it is C, we would be writing G, then C because G always binds to C, A always binds with T, but no T in the case of RNA, we would be writing U. So this is how we will complete the bases in the messenger RNA. You can see here that the bases in these two strands, which are colored by yellow, they are opposite to each other. So if coding strand is given to you, you can find out the basis of the template strand of DNA. You can also find out the basis of messenger RNA. These are very, very simple questions, and you can get full on these type of questions. I hope this was helping. Stay tuned for more videos. Thank you.